my name's Deborah Williamson. I'm a microbiologist and a researcher in Melbourne. And first of all, I'd like to thank the conference organizers for inviting me to present at this wonderful conference. I'd also like to acknowledge the traditional owners of the land on which I'm speaking to you from. And here where I am in Melbourne, this is the land of the Wurundjeri people. And I'd like to pay my respects to their elders, past, present, and emerging. And what I'm talking about today is really the intersection of emerging and re-emerging sexually transmitted infections with genomic technologies. I'll talk briefly through what I mean by genomic technologies and highlight the use of genomics through some case studies, specifically focusing on sexually transmitted Shigella, on gonorrhea, and on syphilis, including some recent work that we've done here in Australia. Now, from really low rates in the 1990s, the 21st century has seen a global resurgence of sexually transmitted infections. What we've also seen are emerging epidemics and outbreaks of what we could term non-classical STIs, and those include enteric pathogens such as Shigella or hepatitis A, pathogens spread by close contact, for example, Neisseria meningitidis, and recently characterized pathogens that can also spread through sexual contact, such as Zika virus. In addition, the emergence of antimicrobial resistance has really raised concerns about increasingly limited treatment options for some STIs, particularly gonorrhea and Mycoplasma genitalium infection. And the factors contributing to sustained transmission of STIs within populations are multiple. They're complex, and we can think of these in terms of the classic epidemiological triad of host, pathogen, and environment. And we recently described some of these in a review article, and they're listed here. Overlaid onto these factors, I guess, are a multitude of socioeconomic and structural variables that can impede access to testing and to treatment. And collectively, all of these factors are really important in driving epidemics of STIs. Now, concurrent with the increase in STIs, there's been a huge explosion in the use of genomic technologies. And what are these technologies? Well, they involve looking at the DNA or the RNA genetic code of a pathogen and analyzing it and then comparing it to other pathogens. And really it can be defined as the study of the content of a pathogen's genetic material. And we can take a range of different approaches here. So for example, we can look at just one area of a pathogen's genome, a single gene or a single region, and that's what we do for HIV genotyping. We can also look at the entire genetic code of the pathogen, and that's what we do for gonorrhea, for example. Or we can look at the genome of one pathogen amongst a multitude of other pathogens, and that's called metagenomics. And we do this for syphilis. And I'll talk briefly through some of these as I go through the talk. Now, when we apply this information to the study of infectious diseases, it's termed molecular or genomic epidemiology. And this information is incredibly useful because it can help us do things like investigate outbreaks, understand the transmission of pathogens, and do things like identify variants of pathogens or vaccine escape mutants. And if this is all sounding a bit familiar at the moment, then that's really because genomic epidemiology is now being used at an unprecedented scale for SARS-CoV-2 surveillance. And we've got things like terms like Delta variants in everyday lingo, thanks to the use of genomic technologies. And I'll come back to the intersection with COVID later in the talk. So the first case study I want to talk about is sexually transmitted Shigella. Now, Shigella is a major global pathogen. It's a major cause of diarrhea, and in fact, it causes bacterial dysentery. And there are two main species of Shigella, Shigella sonii and Shigella flexneri. And one of the hallmarks of Shigella infection is that it has a very low infectious dose. So as few as 10 bacteria can make you really sick. When required, the two antibiotics that are generally used for oral treatment are ciprofloxacin or azithromycin. And in Australia, as in other countries, we have seen a marked increase in Shigella infections over the past decade. And we've seen a drop, obviously, in 2020 for a variety of reasons. And obviously, other countries have seen that, and we've seen that with other infectious diseases too. However, if you just look at these data, if you just look at the incidence data, it doesn't really tell you what the drivers of the increase are. It doesn't tell you what's contributing to that epidemic. So what we wanted to do was to apply genomics to this epidemic to try and identify some of these drivers. So we used whole genome sequencing, and we looked at the genetic code of over 500 Shigella isolates in Melbourne between January 2016 and March 2018. And we integrated the genomic data with some epidemiological data. And that's really where the synergy comes from. Integrating those two data sets gives you really granular insights into what's going on. And so what we identified were 4 main lineages of Shigella sonnei circulating. Now, this is a very brief phylogeny 101. Essentially, this is a phylogenetic tree, and the horizontal lines here mean that isolates are more distantly related to each other. So here, where we've got very few horizontal lines, that's indicative of an outbreak. The isolates are very closely related to each other. So we found these 4 lineages of Shigella circulating, depicted by the different colors there. When we integrated the epidemiological data, what we found was that 3 of the lineages were associated with returning travelers, so people who'd come back with Shigella from overseas. However, this 4th lineage and this outbreak-associated lineage was associated with the MSM community in Australia. We also found that ciprofloxacin resistance was highest in this lineage here, which was associated with travel to South and Central Asia. And concerningly, we found that azithromycin resistance, bearing in mind what I told you, these are the 2 antibiotics that would be used for oral treatment if needed. We found that azithromycin resistance was highest in that MSM-associated lineage. When we looked more deeply at the genomic data, what we found was that the azithromycin resistance was mediated by a gene called MPHA, which was present on a little mobile piece of DNA called a plasmid. And these plasmids can transfer easily between different lineages of Shigella and actually different species of Shigella. We've got this little plasmid, which is causing multi-drug resistance, present in this lineage here, this MSM-associated lineage. A further concern was what happened in the months following this study. So when we revisited the data, what we saw was, yes, we'd seen an increase in this lineage, but we saw an increase in this lineage here in our MSM population. This was the lineage that had initially been associated with travel to South and Central Asia, but this lineage had subsequently acquired that plasmid that I told you about and had become very resistant. So essentially, we have this outbreak of extensively drug-resistant Shigella sonnei amongst MSM, and we described that a year or so ago, resistant to all oral antimicrobials, with over 170 cases since early 2018. And others have found similar findings in different parts of the world. So this MSM-associated Shigella spreading across different continents. So what do we have here? Well, essentially, this is a perfect storm. We've got these dense transmission networks. We've got a pathogen that is highly infectious and a mobile plasmid mediating drug resistance. And one of the hypotheses that we have is that another selection pressure here driving this is the empiric use of azithromycin for many STIs. And that leaves us with relatively few treatment options now for Shigella in some parts of the world. And so, just to summarize there, if we just looked at that graph that I told you about at the beginning, showing the incidence going up, we would have none of these insights. So, but applying genomics shows these really distinct contexts for shigellosis in our settings. We've got this global outbreak of multi-drug and now extensively drug-resistant Shigella. And really, we need to be very vigilant about monitoring this. And this is particularly important because many laboratories now have moved away from culturing Shigella to a purely PCR diagnosis. And without that culture, we can't apply genomics. So I want to move on to talk about applying genomics to gonorrhea now. And gonorrhea and antimicrobial resistance in Neisseria gonorrhea are major public health concerns globally. And resistance to the first-line options, ceftriaxone and azithromycin, has emerged in some parts of the world. And this is really concerning. And along with the potentially severe complications of untreated gonorrhea, mean that gonorrhea is really a major public health problem across the globe. But genomics applied to gonorrhea is not new. It's been used increasingly over the past 5 to 10 years. And some of the pioneers in this work are Jonathan Grad's group at Harvard, Simon Harris's group at the Sanger. And there are a lot of studies now using genomics to look at antimicrobial resistance in Neisseria gonorrhea, to look at how that's evolved, what some of the drivers of antimicrobial resistance might be. However, there are less studies using genomics to look at transmission of gonorrhea within populations. Similar to other settings, and similar to the graph I showed you about Shigella, we've seen a huge increase in the incidence of gonorrhea in Australia. But again, this information doesn't really provide much context about what's driving the increase. So again, we wanted to apply genomics to this epidemic. And we sequenced all isolates in Melbourne from across the entirety of 2017. And really the aim of doing this was to determine the extent and the duration of transmission, including antimicrobial resistance, within and across different sexual networks in our setting, to try and identify possible public health interventions that might interrupt the spread of gonorrhea. And again, we integrated epidemiological data and identified some possible transmission clusters using this data. So basically what we found was that most isolates in the dataset clustered with other isolates, and the median cluster size was 3. So what this is showing, these horizontal lines here are the different clusters across the year. And the numbers across the side represent the different clusters. And you can see that some clusters, for example, cluster 1, cluster 10, cluster 18, cluster 32, they last almost the entire year. And intuitively, you could see here how there might be opportunities to potentially interrupt this transmission if you knew that it was going on, of course. When we integrate the epidemiological data, what we see is that there are distinct associations between clusters and different risk factors. Some clusters, for example, are more heterosexual associated, some are more MSM associated, but we do see bridging of some clusters between different risk groups. We also see that all major clusters involve people living with or without HIV, and we can clearly see here that some clusters are associated with distinct antimicrobial resistance patterns. We were also able to look at clusters containing patients who recently reported having acquired their gonorrhea or having recent sex overseas and possibly acquiring their gonorrhea overseas. And there were 31 clusters in the data set containing patients who reported recent sex overseas. And just over a third of those, the first isolate in those clusters was from a travel-associated case. What does that tell us? Well, it tells us that in some instances, people are coming back from overseas and then new clusters are starting. And immediately, again, you can see some public health implications there for returning travelers. So if people are coming back with either symptoms or having engaged in sex overseas, then that might be an indication for them to go and get tested. And so again, if we just looked at a graph showing an increasing incidence, we wouldn't have any of these insights. So just to summarize that section on gonorrhea, resistance in gonorrhea, I hope I've shown you, is concentrated in distinct populations and networks. If you just lump all of the data together, you lose that granularity without the genomics applied, integrated with the epidemiological data. And really, when you do integrate those two data sets, you can start to untangle some of the complex epidemiological and behavioral factors that drive transmission. And that's when you can have precision interventions to interrupt transmission. The final case study I want to briefly mention is how genomics is being applied to Treponema pallidum, so the causative agent of syphilis. Now, Treponema pallidum is extremely tricky to work with in the laboratory. It's not like gonorrhea, it's not like Shigella. It doesn't grow on an agar plate. It doesn't grow in conventional culture, and that makes genomics very tricky to do on Treponema pallidum. Coupled with that, there isn't very much in a clinical sample, so we've really had to adapt our genomic approaches here. And we apply metagenomic approaches to look at Treponema pallidum, and we combine it with something called target enrichment. And what that does is basically it pulls out all of the sequences in a clinical sample that look like Treponema pallidum, and then we apply sequencing to that. Because this process is quite labor intense and it's actually quite expensive, there are really only a handful of studies applying genomics to Treponema pallidum at the moment. And this is the most recent one, and this is a study coming out of the Sanger that we were pleased to be able to contribute to. And essentially what this study shows is that, far from being an old medieval disease that hasn't changed much, there's actually been a lot of genomic movement in the Treponema pallidum population. And this study is a really nice study, which essentially shows that Treponema pallidum has two major lineages, the SS14 lineage and the Nichols lineage, and they split sometime in the 16th or 17th century. What we've seen in the 21st century are these emergence of major dominant lineages, which look quite different from the older lineages, characterized by a dramatic population expansion concurrent with the increase in other STIs, and again macrolide resistance, or resistance to azithromycin. And so the modern syphilis epidemic that we have at the moment, that isn't just one lineage, it's multiple lineages which are spread across different continents. And again, we wouldn't have these insights if we hadn't applied genomics to the investigation of the syphilis epidemic. But really, I do think that we're just scratching the surface about what we know about Treponema pallidum, and genomics could really give rise to much broader insights into pathogenesis and potentially vaccine development for syphilis as well, because we'd be able to look at things like antigenic variation in different lineages. Okay, so I mentioned that genomic epidemiology has really come into its own over the past year with the application to COVID-19, and I think that we can learn a lot about the way that different countries and different groups within countries have really mobilized to freely share data and information relating to the spread of this infection. And uploading data to publicly available databases such as NextStrain has really made genomics so much more accessible. Another thing that the COVID-19 pandemic, I think, has catalyzed is the implementation of genomics into real-time public health responses. And so, we've seen that at a population level where we've monitored the emergence of the Delta variant. People have based policies on the emergence of this variant. And we've used this in our local setting to look at the emergence of COVID-19, and really in a low prevalence setting, hopefully still in Australia, we've really used it to try and differentiate imported cases of COVID-19 versus local transmission. And this figure essentially shows that the early stages of the COVID-19 pandemic in Australia back in early to mid 2020 was characterized by multiple introductions from overseas. But we had this second wave in Melbourne in the mid to late last year with a very severe and prolonged lockdown. And you can see here that there was really what caused that was a breach in the hotel quarantine system and then spread throughout the community. And so genomics really came into its own there. And I think if we were able to mobilize in that way, you know, and use, take genomic data and apply it to the investigation of STIs, then, you know, we really would be able to tailor our public health interventions better. Finally, I want to finish by acknowledging the years of work that's been done in the HIV molecular epidemiology space. And HIV molecular epidemiology is a talk in and of itself. And in particular, this audience and this community has laid important groundwork and had important discussions about the responsible use of molecular epidemiology. And this is something that's relatively nascent, I think, in the STI genomics field, but it has to be addressed as the technology becomes more widely available. Finally, to conclude, I hope I've shown you that rates of established STIs are really approaching levels we haven't seen in decades. Genome sequencing is this powerful tool that we have now that can monitor the emergence and spread of infections, including STIs, across the globe. And the use of genomic technologies, the responsible use of genomic technologies, may enable targeting of public health interventions. And, you know, we really do need much more sophisticated approaches to the prevention and control of STIs. There are a large number of people to acknowledge. I particularly want to acknowledge my collaborators at Melbourne Sexual Health Centre. And I'd like to thank you very much for listening. Thank you.